All right. Thank you so much for the invitation, Dr. Meekins. When you reached out to me to give this talk on a live stream, I was super excited to try it out. And so I'm looking forward to any questions, and especially if you have questions about terminology that I'm using and things like that, please don't hesitate to ask because I'm happy to slow down and really dive into the basics and the background of what we work on in my lab. And so the title of my talk is Let's Talk About Flux, Baby. And we're really focused on making dynamic autophagy flux measurements for different biomedical applications. And if you are not a 90s hip hop fan, if that's not your jam, hopefully there are some other music groups scattered throughout this talk and some Easter eggs that you will enjoy instead. Um, okay, so to give you a big an overview of what my group works on at UC Davis, you learned already that I have a joint appointment in microbiology and in chemical engineering. So we really work at the intersection of these two disciplines in my group. And on the microbiology side or the virology side, we are really interested in understanding how viruses turn cells into tiny little microscopic viral factories. So if you zoom all the way into a single cell in the human body, what does a virus have to do to make more of itself? And we use quantitative approaches and chemical engineering principles to study these types of phenomenon. And then on more of the engineering side of things, we also spend a lot of our time building tools to study virus host interactions more quantitatively. So my entire lab is really focused on studying virus host interactions, and we really want to do it in the most quantitative manner possible. Sometimes we find that those tools don't exist, and so we have to take a step back and build those tools. And so today's talk is really on this engineering side of the coin that we work on, which is taking that step back and building tools to study a specific phenomenon that occurs in cells. And that's a phenomenon called autophagy. So autophagy it's a compound word that made up of auto or self and phagy, which means eating. So it's a self-eating process that is used by all of the cells for all the way from yeast to humans to recycle cellular building blocks. And this is a cartoon diagram of what that pathway in cells looks like. So there are these specialized organelles that are called phagophores inside of our eukaryotic cells, and they grow to encompass the cellular contents. So the stuff that's just floating around in the cytoplasm. So that might be proteins, it might be RNA, it might even be entire organelles. Like here, I'm showing you a mitochondrion that's getting enveloped by a phagophore. These phagophores grow and eventually close in on themselves to form autophagosomes. Those autophagosomes then fuse with lysosomes. These are other organelles inside the cell that then form these autolysosomes in this fusing process. And so the lysosomes contain special enzymes and a low pH environment. And that low pH environment with those special enzymes help break down all of these cellular contents. So that might be damaged proteins or organelles. And out on the other end, what you get are amino acids, fatty acids, and energy. And so the reason my group got interested in autophagy is because it's dysregulated in a lot of different diseases. So it's commonly dysregulated during aging, neurodegeneration, cancer, but also during microbial infections and especially during viral infections. And that's how my group got interested in autophagy. So to give you an idea of how autophagy is manipulated during viral infection, typically autophagy is considered to be antiviral. So autophagy is often used to inhibit the virus. And that's because in addition to degrading cellular proteins and organelles, this entire pathway can also be used to encompass entire virions or viral particles so that the virus doesn't replicate as well. And this pathway is so antiviral that some viruses have evolved mechanisms to inhibit this pathway. And what's really interesting for my group is we study Zika virus. And Zika virus figured out a way to rewire this entire pathway because autophagy actually helps the virus replicate. And the virus actually has mechanisms to induce autophagy so that it can replicate better. And so this rewiring of the autophagy pathway was really interesting to us. And we were interested in specifically, how does the virus do this? If you look at a single cell and you zoom into that molecular level detail, how does this virus rewire this complicated pathway that's so important for cellular homeostasis? And so one of the things that I had discovered previously is that Zika virus has a lot of protein interactions with proteins that are involved in this autophagy pathway. So here I'm showing you a cartoon summary of some of those protein interactions, where the gray squares are the Zika virus proteins, and these blue circles are the autophagy proteins from human cells. And where you see a line connecting them means that we identified a protein interaction between this viral protein and this cellular protein, this host protein, in other experiments that I'm not going to go into the details of. And so because we had all of these protein interactions with these autophagy proteins, we got really interested in studying autophagy and studying how Zika virus manipulates the autophagy pathway. And so we wanted to understand whether specifically any of these protein interactions are responsible for the behavior that we see in general. But what we realized is that we needed to take a step back and think about how we were going to measure autophagy in Zika virus infected cells. And the reason for that is we wanted to put our chemical engineering hats on to solve this problem. And we looked at this pathway and realized, like others in the field, like Ben Luce, who published a similar paper in 2014, noticed is that autophagy is just a series of reactions. And so you can describe the formation of autophagosomes as a reaction or reaction one. You can describe the fusion of autophagosomes and lysosomes as reaction two to form autolysosomes. And then you can describe the turnover of autolysosomes to form free amino acids and fatty acids as reaction three or R3. And so as chemical engineers or as chemists, you might perform a mass balance on these reaction intermediates, as one might do, to calculate the steady state flux through this autophagy pathway. And so here I'm just showing you some simple ordinary differential equations that perform that mass balance on these reaction intermediates. So here we have autophagosomes, or AP. And so we can say that the accumulation of autophagosomes with respect to time is equal to the rate of their formation minus the rate of their consumption through this fusion step with lysosomes. And we can say the same for autolysosomes. The rate of accumulation of autolysosomes is equal to the rate of their formation through this fusion step and the rate of their consumption through turnover. So this is not rocket science. This is stuff that we cover in undergraduate kinetics. And we can say that at steady state, these rates of accumulation are equal to zero. And so then if we can figure out the rate of one of these steps, we can figure out the rate or the flux through this entire pathway. And so what biologists have been doing for a long time, but not very quantitatively, is doing exactly this using chemical treatments. And so what they do is they treat the cells with a drug called bifilomycin A1, and this inhibits the second step or the second reaction in this series. So basically, we have a chemical that allows us to set the second rate, R2, equal to zero. And then if we have a way of measuring this accumulation of autophagosomes, if we can see how this increases over time because they're no longer being consumed, then we have a way of calculating what that steady state flux through the autophagy pathway is. And that's exactly what people have been doing, mostly semi-quantitatively or qualitatively at best. And what a graduate student in my group, Nitin Bisavathuni, decided to ask as his thesis project was, can we actually measure the autophagy rates in a non-steady state system? And the reason for that, or the reason we were motivated to do that, is because virus infection is a dynamic process. And if Zika virus, for example, is rewiring this entire pathway, wouldn't it be interesting if it was doing it in a way where it was actually creating a non-steady state between R1, R2, and R3, and we could actually capture that so that it could induce the parts of the pathway that were valuable to its replication and also inhibit the parts of the pathway that could potentially be detrimental to its replication. And so Nitin decided to build a system that would allow him to do that. And so this is the point where we took a step back and said, let's take some time to build the system and let's test it out with some very well-established manipulations of the autophagy pathway instead of working with viruses, okay? And so this is the approach that Nitin decided to use. What he realized is that an instantaneous rate analysis, which is what chemists and chemical engineers often use to characterize reactions, would allow us to capture these non-steady state rates. And so we're using the same mass balances on autophagosomes and autolysosomes. And we're also using the spifilomycin A1 to inhibit the second reaction and set R2 equal to 0, just like before. The major difference is that what Nitin realized is that if we had an ability to measure this rate of accumulation of autophagosomes with respect to time over a very short period of time, and then we could add bifilomycin to the system and measure this rate of accumulation after we add bifilomycin, we would actually be able to uncouple R1 from R2 and R3. We would no longer have to assume that they're equal because we no longer have to wait for this rate of accumulation to be 0. If we can actually measure this before we add bifilomycin, we can extract all of these rates independently. And so that's exactly what Nitin set out to do. And the reason no one had done this before is because it's actually really technically challenging to make these measurements on a very short time scale on the order of 5 to 10 minutes. Okay. And so the system he used, okay, before I go into the system, I forgot, I should show you graphically what we're talking about. So in terms of the previously established methods to measure the steady state flux through the autophagy pathway, this is graphically what it looks like. So if we're looking at the autophagosome number per cell over time on the x-axis here, then at steady state, this number is not changing. And when we add bifilomycin, we can measure the slope and calculate the flux. That's exactly what we were doing before, pretty straightforward. But in a non-steady state system, this autophagosome number per cell, it's changing over time. It's no longer constant. And so now if we can measure the slope before we add bifilomycin and also after we add bifilomycin, now we can extract R1 without assuming that it equals R2 and R3. And so that's exactly what he set out to do. And the system that Nitin decided to use to track autophagy reaction intermediates is this tandem reporter system developed by Tanita and colleagues in 2014. And so in the system, we engineer cells with a fluorescent protein. And so the protein that we're really interested in is this protein called LC3. And it is a protein that labels phagophores, autophagosomes, and autolysosomes. It's present on all of these different organelles. And we engineered cells that express a fluorescent version of this LC3 protein It contains a red fluorescent protein called MK2 and also a green fluorescent protein called fluorin. And fluorin is a really interesting protein because it is pH sensitive. So it is quenched in a low pH environment. And so autophagosomes, phagophores and autophagosomes have a neutral pH. And so they fluoresce both green and red. And so they appear yellow under a microscope. But autolysosomes, because of the fusion with the low pH lysosome, these have a low pH environment. And so the fluorin fluorescence gets quenched and autolysosomes appear red only. And so by tracking these different spots inside cells, we can determine whether they are both red and green, and so an autophagosome or a phagophore, or whether they are just red and therefore just an autolysosome. And so Nitin engineered these cells and he then set up a live microscopy system. So this is a system that is specially built to take images of human cells that are incubated at standard, human body conditions at 37 degrees Celsius with the right amount of CO2 and humidity. So there's a specialized incubation chamber that this microscope sits in. And then it's automated to take images every 5, 10, or however often you want at the exact same position so that we can stitch these images together and create these time-lapse videos. So here I'm showing you an image of these engineered cells over a 15-hour experiment in this case. And what you can see, these large circles in the center are the nuclei of the cells. And then these tiny little green dots, those are the autophagosomes or the phagophores. And so here I'm just showing you the green channel, but there's a similar video that we can also take of the red channel. And then what we do is we take these images, and Nitin developed an automated image analysis pipeline to take these images and quantify the number of cells, which are indicated by these yellow dots, where he identified these black areas that are the nuclei of each cell. And he quantified the autophagosomes and the autolysosomes in each cell with these magenta and red dots. And so he did this for thousands of pictures over several hours of each of his experiments, and developed a system to track these autophagy intermediates over time so that he could measure these rates of accumulation. Okay, and so then he went on to actually test out the system. And again, we decided to take a step back. Instead of focusing on viruses, which is how we originally got interested in this pathway, we decided to use some really well-characterized drugs that manipulate the autophagy pathway. And so the first drug that we tested was called rapamycin. And so rapamycin acts on the very first step in the autophagy pathway. And it actually acts to induce the autophagy pathway. So when you add rapamycin to cells, you should get more flux through the pathway, or that's what we expected. And so our first question was, what happens to autophagy rates when we induce autophagosome formation with rapamycin? Do we actually observe this increase in flux? And can we resolve the time period over which that happens? And so Nitin performed these measurements over a 15-hour time course where he quantified autophagosome and autolysosome number over time, and then performed a lot of calculations to extract rates for the first step, R1, is what I'm showing you here. And I'm showing you a gray line, which represents untreated cells that have just a carrier added to them. So it's DMSO, so they have no rapamycin. And then the teal line here represents a really high concentration of rapamycin of 1,000 nanomolar. So this is kind of like a sledgehammer. If you add this to cells, you should see a large induction of autophagy. And so what was really exciting to see is that we did indeed see increases in R1 that were significantly higher than the cells that remained untreated. But what was really exciting to us is that we were able to resolve these differences in less than 10 minutes. So the earliest measurement we were able to take was right at 10 minutes. And we already see a huge increase of almost doubling our initial autophagy rate of R1 at this 10-minute time point. And so we anticipate that if we were able to make these measurements on an even tighter time resolution, we would see differences even at 5 minutes. And so this was really exciting because in the literature, the shortest timescale measurements people have really taken for autophagy measurements, as far as I know, are on the order of hours. And so getting this type of time resolution is really exciting. And I'll talk a little bit about why that might be the case in a few minutes. We were also able to extract rates for the second and the third steps in the autophagy pathway. So this is R2 and R3. And again, compared to the basal or the untreated cells, we observed significant increases in these rates. What was really exciting to us was that we did not see these significant increases until 30 minutes after treatment, which I'm showing you here. So at 10 minutes post-treatment, our first measurement of R2 and R3, those rates were indistinguishable from our untreated cells. And if we go back to our understanding of how rapamycin works, we know that it acts on this first step in the pathway. And so what we're observing here is a latency in the system that we can actually resolve using our method. Just a moment. I've got to give my son more time on his iPad. Sorry, folks, this is real life as an academic parent. All right. Thanks for your patience. So we were able to resolve all three rates and resolve the time evolution of these rates to see that R1 increased first, and then R2 and R3 caught up after a short latency period. We could do this for a lot of different concentrations of rapamycin. So here, what I'm plotting are the initial rates that we measured. So the rates that we measured at 10 minutes post-treatment for the first step in the pathway. So this is the R1, that first reaction. And I'm showing you that initial 10-minute measurement for R1 at different concentrations of rapamycin on a logarithmic scale. And so what I hope you can appreciate is that we get this nice sigmoidal response for our initial rates, and we can actually use them to estimate kinetic parameters for rapamycin. And here we could estimate a Vmax, a Michaelis-Menten constant, and a Hill coefficient for this process. And the reason we think that this might be really exciting, these initial rate measurements on a really short time scale, is that they can actually give us insight into the mechanism of action of a drug. Because most of these measurements that people are making on autophagy flux that happen after a couple hours might already take into account feedback mechanisms that the cell has kicked in to control this perturbation that it is experiencing. And you can even see that here, where the rate increases and then it decreases over some period of time. And that's also true of R2 and R3. So the flux increases initially, and then it slowly tapers off. And that's not a result of a short half-life of rapamycin. Rapamycin is actually quite a stable drug. And so what this reflects is the cellular feedback response that you're seeing at these later time points. And so these early time point measurements help us deconvolute these feedback mechanisms from direct mechanisms of action. All right, so we also wanted to test out other drugs, not just rapamycin. It worked great in our hands, but we wanted to test out some other things. So we tested out something called Wortmannin, which also acts on this very first step of the autophagy pathway, but it acts to inhibit autophagosome formation. And so we wanted to test whether we could actually measure this inhibition using our new approach. And so we performed the same experiments where we would treat with Wortmannin and then measure these rates individually. And now I'm showing you again the rates R1, R2, and R3, each of these three steps for the untreated cells or the basal cells in gray. Now they look up higher on the graph or on the plot versus the colored bar, either turquoise, orange, or magenta, represent the rates after treatment with Wortmannin. And again, at very early time points for 1 micromolar Wortmannin treatment, so that's a very high concentration of Wortmannin treatment, at very early time points, even just 10 minutes post-treatment, we're able to resolve a huge decrease in the first rate, the rate of autophagosome formation following Wortmannin treatment, as we would expect. And then the cells actually quickly recover. And we believe that this is because of the short half-life of Wortmannin in cell culture media. But when we look at these other rates, the rate of autophagosome lysosome fusion and also the rate of autolysosome turnover, we see that similar to rapamycin, we don't observe a difference at 10 minutes post-treatment, but at 30 minutes post-treatment, we do see a significant decrease. So again, we observe that the cells have a latency in the pathway, and it takes about 30 minutes for the second and the third reactions to catch up in terms of the increase or the decrease in the rate in this case. All right. So now that we've got this really exciting method to measure autophagosome dynamics and really measure individual rates in the autophagy pathway over time, we thought that this might be an exciting way to figure out, first of all, how much cargo actually gets degraded. So not only from a virus perspective, where we think the front end of the pathway might be induced and the back end of the pathway is getting inhibited. If you're thinking about developing therapies, you might actually want to fine tune the total flux through the pathway and make sure that this last step isn't a rate limiting step, and that autophagy, the contents that's getting consumed by these autophagosomes, is actually getting degraded, if that is what's going to give you the therapeutic effect. And so we wondered, do the temporal differences between R1, R2, and R3 matter? And could we actually calculate that? And in fact, by having these individual measurements for R1, R2, and R3, we can resolve the total degradative capacity of the pathway by taking the area under the curve of this third rate, the rate of autolysosome turnover. So if we've got this rate evolution over time for the third rate or the third reaction in the series, simply taking this integral or the area under the curve can give us the overall cargo degraded and allow us to compare different treatments over time. And so in the interest of time, I'm not going to spend a lot of time talking about how we've used this, but it is another tool or another metric that we can use to be more quantitative about our measurements of the autophagy pathway and flux through this pathway. All right. So to summarize up to this point, we've developed this quantitative systems autophagy framework, and we've tested individual drugs on this pathway. I've shown you some of the data related to that. We've also tested drug combinations using this methodology. We've also tested other biologically relevant conditions like nutrient starvation. This is a condition that happens often during cancer because complex tumor environments are often nutrient deprived. And so autophagy is manipulated quite a bit by tumors and the tumor microenvironment to help provide energy to the tumor. And so we were able to show that nutrient starvation conditions can also be used to resolve differences in autophagy rates. We've quantified the total degradative capacity, like I showed you, by taking the area under the curve for R3. And moving forward, we're really interested in applying this technique to understand how viruses perturb autophagy. That was the original question that motivated our work. But ultimately, we think that this is a tool with a lot of biomedical applications. And so one of the things that we've been thinking a lot about now is whether this type of tool can be applied to high throughput drug screening, because a lot of companies are looking at autophagy manipulators to treat cancer, neurodegeneration, and infections. So what I want to spend the rest of the time talking about is our efforts in high throughput drug screening. But in the meantime, I'm happy to pause here and to answer any questions that might have arisen. Oh, that's a great question. Um, so for this type of process, there are side reactions. There most certainly are side reactions. We don't have a good way of monitoring them, unfortunately. And so it essentially goes into our assumptions, right? That you have to acknowledge that there may be side reactions. There are other pathways that use the organelles that are involved in autophagy as well.
Okay.
Um, and and so what we are monitoring may not be truly just autophagosomes and the autophagy pathway. It may include other pathways as well.
Okay. Do you have a sense of, like, I mean, a rough idea of how much of an effect it is, or is it one of those things that is hard to quantify at this point?
At this point, we don't have the tools to quantify it that well. And what I will say is that it's going to be really context dependent. So it's
Sure.
going to depend on the type of cells that you use, what
Okay.
kind of tissues they came from, and things like that.
Okay. Interesting. Let's see. Does the motion of the cells ever cause any problems in terms of measuring the fluorescence?
Oh, that's a great question. That is a real technical challenge for us. So we use adherent cells, so they stick to the surface of our
Okay.
culture plates. And we have to do that because if they're floating around, then we can't really track them, and they
Mm-hmm.
might be in different focal planes, which makes it really
Oh,
challenging.
yeah. Okay.
Because the type of imaging we do, um, you can certainly take images in different Z planes.
Mm-hmm.
People do that all the time. But for us, we're really interested in getting more information about more cells
Yeah.
than getting thorough information about a few cells. And
Okay.
so these are some of the caveats that you do have to consider when you're developing one of these systems. Are you going to go for really fine grained data of a small set of representative cells,
Mm-hmm.
or are you going to try and collect data on as many cells as you can? Um, so not all cells are equally adherent either. And
Oh.
so we've started applying some of these measurements to better models of cancer because we're really interested in how autophagy flux is manipulated in cancer in some of our collaborations.
Okay.
And so in those experiments, cancer cells, they move around a lot because that's actually how they grow, right? That's how they get out of that nutrient starved microenvironment and
Oh.
get into new areas of the body like
Yeah.
metastatic cancer, right? So a lot of these cancer cells that people work with in research, they move around quite a bit, even though they kind of stick to the cells or stick to the plates.
Got you.
And so it's a real challenge.
Yeah.
And they'll grow on top of each other. Um, that's a whole mess
I
too.
hadn't even thought about
There
that.
are, yeah, there are ways to overcome that, but you have to be really committed to making it work.
Hey, fair. This is why I don't do anything related to biology.
Mhm.
Let's see. The last one is what drives the lag time of the R1, R2, and R3
Oh,
variables, I
that's
guess,
a
that are
great
measured?
question. Yeah. So why is there a latency? There must be something that's rate limiting.
Mm-hmm.
And we hypothesize that it has to do with the R2 step and that the R3 is just a consequence
Oh,
of
okay.
being behind R2.
Makes sense.
Because if we look at the dynamics, the R3 runs in tandem with
Oh, okay.
R2,
Gotcha.
right? There's no additional delay. If you overlay those curves,
Okay.
which I didn't show you,
Mm-hmm.
but if you do overlay them, then what you will see is that R2 is really driving that latency.
Gotcha.
Um, we thought that there might be like a limitation in the number of lysosomes in the cell. So
Mm-hmm.
maybe, we increased R1. There were so many autophagosomes that there weren't enough lysosomes to actually fuse with anymore. And
Oh.
that this was a limiting reagent essentially.
Mm-hmm.
But we quantified it, and that was clearly not an issue.
Okay.
It's not like we couldn't find any lysosomes anymore
Yeah.
in the cells,
Yeah.
that they had all been soaked up.
Interesting.
Um, yeah. And
Okay.
so we think that there might be some protein,
Mm-hmm.
or maybe there's the energy required for this fusion step
Mm-hmm.
is limiting.
Okay. That would make a lot of sense. Okay.
Yeah. But
Cool.
that's why like having these types of temporal measurements can be really valuable,
Oh
because
yeah.
you can start figuring out what's rate limiting.
Mm-hmm. Yeah, I
Yeah.
mean, yeah,
It gives
it's really
you
cool how you're able to separate all the steps.
yeah, it actually gives you a place to look, right?
Mm-hmm.
So
Mm-hmm.
um
That's the first thing you got to know what you're looking for first. So yeah, that makes a lot of
yeah.
sense. All right,
Hmm.
cool. I think that's all the questions for right now. So I'll let you keep going.
Okay. All right. So we wanted to take this approach and apply it to high throughput drug screening, right? That was one of our larger goals after publishing this work last year. And so one of the issues that we had is that these rate measurements that Nitin is making, they are not scalable at all. So these are incredibly labor intensive measurements that take, first of all, super long days. If you're making some of these measurements, he's in the lab late at night. Um, and it's like an early morning all the way to maybe 2 or 3 a.m. at night. And then on top of that, if you want to test a bunch of different conditions, it's just not going to work. And so if you think about if you have a large drug library of thousands, 10,000s, hundreds of thousands of chemicals, and you want to identify which drugs you want to use because they modulate these rates so that you can precisely regulate autophagy and turn the knob on the autophagy pathway to either increase it or decrease it therapeutically. Well, this method is not going to work on 100,000 chemicals. And so, what Nitin realized is that we really need to develop a higher throughput method to screen these larger drug libraries. And so, what he thought about was using image based profiling, and the idea that he has all of these images, and an image is truly worth 1,000 words. And he's only using a small amount of the information in these images to make these rate measurements. But what other information is in these images to actually figure out how autophagy is manipulated, even if it may not be as intuitive as measuring autophagosome and autolysosome number? And can we use all of that information to then narrow down the number of drugs that we need to test in a more rigorous way using these more labor intensive measurements to regulate autophagy precisely? And so this is something that Nitin started towards the end of his PhD. And he recently graduated. And so this is being continued by some other members of the lab, an undergraduate, Ishan Tilakaratne, a master's student, Ritika Gangaraju, and a PhD student, Neil Adia. Okay, and so to explain what we mean by extracting all of these image-based features, or using image-based profiling to understand how autophagy is being manipulated. First, I want to tell you what image-based cellular features are. So basically, each image has a wealth of information in it. And so we can categorize them into three different types of features: shape features, intensity features, and texture features. So shape features have to do with physical geometry of either the cells or the dots that represent the autophagosomes and autolysosomes that I showed you in that video earlier. And so examples include like the radius of the cell or the dot, the area, what the perimeter looks like, the roundness of the cell. You can also calculate Zernike moments, things like that. For intensity, we're talking about the intensity of the fluorescence. So you can calculate the mean intensity across an entire cell, and it might be higher or lower. You can calculate the max intensity, the minimum intensity, median, etc. And then there are also these texture features. And this has to do with how the signal is distributed, how that fluorescence signal is distributed. So here I'm showing you three different examples, and they all have the same overall intensity because they have the same number of black and white pixels. But they're all distributed in very different ways, and they mean very different things to us as a result. And so by taking these texture features, you can understand how the cellular material is organized inside a cell. And the features that we keep track of are contrast, something called entropy, variance, energy, and correlations between different features or different fluorescence channels as well. All right, so Nitin built a pipeline that would take all of these images that he had collected over several years of research, in an effort to optimize this pipeline that he built that I showed you earlier. And he built out a system with Ishan, an undergraduate student working with him, to extract about 500 features for each cell in an image. And he had a way of actually stitching them together over time so that he could track how a single cell's features evolve over time. And so that is what I'm showing you here. It's the automated pipeline that he uses to track and extract features at a single cell level, which he built in Python using Napari and btrack, among other things. And so here I'm showing you a video that shows how the cells are segmented so that individual cells can be monitored over time. And the puncta are identified. Those autophagosome and autolysosome dots, those fluorescent dots are identified over time. And then that's from a single image on the left. And then on the right, what I'm showing you is one of his time-lapse experiments where those cells are then tracked frame to frame, and you can monitor the trajectory. So here you can see a little squiggle, and that squiggle represents the trajectory of that cell from frame to frame over the course of this experiment. And so all of these features are extracted from this cell and all of the other cells in this frame or in this field from frame to frame and stitched together into a large multidimensional matrix of features that are time resolved. All right. So then Nitin wanted to apply this pipeline to rapamycin and Wortmannin, the two drugs that I showed you earlier. And remember, rapamycin induces autophagy flux, and Wortmannin inhibits autophagy flux. And I showed you data supporting that earlier. And so he wanted to figure out whether he could actually resolve these differences in rapamycin and Wortmannin treatment without making those rate measurements. And this is some data showing that he can, in fact, identify features that are significantly different in the two treatments. So here each dot represents a feature that was extracted from those different images that I showed you. And what we're looking at is the median z-score of that feature relative to untreated cells. So here, the features are compared from rapamycin treated cells to untreated cells. And so if the median z-score is 0, that means that there was essentially no difference in that feature between rapamycin and untreated cells. And so a positive deviation means that feature increased following rapamycin treatment, and a negative deviation means that feature decreased following rapamycin treatment. Excuse me. And then on the y-axis, we're just looking at the significance of that increase or decrease using a log transformed FDR score. And so we can see that there are a lot of things that increase significantly and decrease significantly for rapamycin treatment and for Wortmannin treatment. The question is whether those things are different or not, or whether everything's changing kind of the same, even though these true treatments do very different things. And in fact, if we dig into the data and what these features are, there are some things that make a lot of sense. So first of all, if we just look at autophagosome number, that's one of the features that Nitin continued to extract. He was extracting it originally with his more involved rate measurement pipeline, but now with this image-based profiling pipeline, he's still extracting those autophagosome numbers over time. And for rapamycin treatment, those autophagosome numbers significantly increased following treatment compared to the untreated cells, which is what we would expect for something that induces the autophagy pathway. And versus Wortmannin, those autophagosome numbers significantly decreased following Wortmannin treatment. Again, something that we would expect for a proper inhibitor of the autophagy pathway. The other thing that made sense was that the mean intensity of the cells decreased for rapamycin treatment compared to untreated cells. And why would the mean intensity decrease? So the mean intensity refers to the fluorescence intensity of those cells. And that fluorescence intensity is coming from that LC3 protein that is labeling all those autophagosomes and autolysosomes. But if autophagosomes are being produced, then why is the intensity decreasing? It's because they're also being degraded. When those autolysosomes get turned over, the LC3 protein also gets degraded along with the other proteins inside those autolysosomes. And so while the total autophagosome number increases, the total amount of LC3 protein and therefore the mean intensity of those cells actually decreases when you induce the autophagy pathway with rapamycin, as we expect here. And the exact opposite happens with Wortmannin treatment. The mean intensity actually increases. Even though we're producing fewer autophagosomes, the fact that the autolysosomes are no longer being turned over as much means that fluorescence from the LC3 actually increases. And so this was really exciting because it was a nice sanity check for our overall approach. But then we also wanted to take all of these other features and figure out whether those could actually be predictive of the overall treatment that we were giving. And so I'm just going to show you a brief overview of what we've done, though there is more that has been pre-printed since then, beyond what I'm showing you here. But here, what we're doing is we're using UMAP clustering to figure out whether the features that we've extracted can actually be used to identify Wortmannin treated cells versus rapamycin treated cells without making any measurements of rates. And so before treatment, the Wortmannin treated cells, the rapamycin treated cells, and the DMSO treated cells, so that's basically like untreated cells, all of those cells kind of clustered together in a giant mix. That's exactly what you would expect because they're kind of all treated the same. They're untreated. But after treatment, and this is again time resolved to include all of the data over 15 hours of measurements, each of these dots represents a single cell that we measured following either rapamycin treatment, Wortmannin treatment, or DMSO treatment over a 15-hour period, and extracted all of these features. And what I hope you can appreciate is that the Wortmannin treated cells all cluster pretty much similarly. The rapamycin treated cells also cluster mostly to themselves, and the DMSO treated cells cluster kind of in the middle with a little mixing with the rapamycin treated cells. And so here, this is just the tip of the iceberg of what we hope we can do with image-based profiling to help resolve different types of chemical treatments or other types of perturbations on autophagy on a higher throughput scale. And so moving forward, we're really excited to, first of all, continuing to develop this image-based profiling pipeline and apply it to drug screening, but also expand beyond drug screening. So we're obviously going to go back to Zika virus infection, what got us interested in this project to begin with. But autophagosomes are really just one aspect of the cellular response to different types of perturbation, whether that's a drug or an environmental perturbation like nutrient starvation or virus infection. And so wouldn't it be great if we could also build similar tools to track other very important organelles inside the cell, including the endoplasmic reticulum, where a lot of biosynthesis occurs, and mitochondria, which regulates so much of the metabolic potential of cells and end up being really important in cancer, neurodegeneration, and virus infection. All right. So with that, I'm just going to make a pitch that if you got excited about what you saw today, I highly encourage students to consider graduate research or postdoctoral research at UC Davis. We've got a number of highly ranked graduate programs. We've got unique resources at UC Davis, including a national primate center, one of only a handful in the entire country, a medical school, and one of the best vet schools in the world. We've got a very collaborative student environment. It's affordable, and I will highlight it's affordable for California, and we do have a new graduate student contract. The students are unionized, and we're close to mountains, beaches, and cities. So whether you like to ski, surf, or skateboard, hopefully you can find your thing at UC Davis. With that, I'll acknowledge the group of wonderful researchers that I get to work with every day in the Shah Lab, and especially Nithin Bisa-Bathuni, who was the brains behind this entire project. He really thought it up and pushed it forward. And then also Neil, Rithika, and Ishan, and Vardhan, who are working on it now. I'll also thank the Keck Foundation for their funding, and I'm happy to take more questions.
That's really cool stuff. So I think we got just one more question here. So you get any information from time resolved versions of those z-score tabulations? I think that's the one that you showed where it had the positive and negative deviation. And I think the one you had was 3 hours. Is that right?
Yeah.
Okay. Do you get any additional information, like if you go from 1 hour to 3 hours to 10 hours or something like that?
Yeah. So what we see is that there are dynamics in how many features change and when they are changing.
Okay.
So typically we see a huge increase in the number of features that are changing early on,
Okay.
and then it might slowly go back to normal.
Okay.
And sometimes we see like that there's an increase, it goes back down, and then it starts actually coming back up. So there might be some sort of oscillations that the cell is experiencing that we're just starting to capture. Yeah.
That's very cool. Okay. Nice.
Yeah, I mean, from a controls perspective, there's a lot of exciting stuff.
Yeah.
Here.
Absolutely. Yeah. No, this is really cool work. Like I said, I had no
Mhm.
idea what was coming, but you did a really nice job of explaining it, and
Mhm.
yeah, it's really really interesting the way you're digging into the sort of like it's fundamental work, but the applications are very clear as well. So this is really really neat stuff, and I appreciate you sharing it with us.
Well, thanks. I really enjoyed it.
Good,
And
I'm
thanks for
glad.
all the great questions.
Yeah, absolutely. So I think if there's no other questions, we'll say thank you again to Dr. Shah. I really appreciate it. Thank you to everybody for coming out, and when we have another talk, I hope you will all come back out and join us.
All right.
All right, thanks very
Well,
much.
take care.
Yeah. All right, hang on one second. We'll talk very briefly.